Okay, we'll go a little more into detail about the male reproductive system, some aspects of mitosis and meiosis, what's involved in the development during spermatogenesis, which is an integrated process in development. It involves mitosis, which typically is referred to as the division of somatic cells, and meiosis, which specifically refers to the division of the gametes. In the process of mitosis, the spermatogona undergo cellular division throughout their adult life. You have one daughter cell, and each division will remain in place while the other ones are pushed forward through the lumen of the seminiferous tubules. Now, if you remember the seminiferous tubules, what their relationship is with the testicles, they will be the portion of the anatomy where the sperm will continue to develop and then will be pushed on into the vas deferens, which is the ductal system that will lead through the male reproductive system. The displaced cells will then differentiate into primary spermatocytes, which are prepared to undergo meiosis. So meiosis is a special form of cell division. It involves gamete production. Gametes in the humans contain 23 chromosomes, half the normal set. As a result, you'll see a fusion of the nuclei of the male gamete and the female gamete, which will produce the normal number of chromosomes, which is 46. Now, in the seminiferous tubules in the male reproductive system, meiotic divisions will begin. The primary spermatocyte will then produce spermatids. These are undifferentiated male gametes. In the process of spermiogenesis, you see these spermatids will then differentiate physically into mature spermatozoa, which are among the most highly specialized cells in the body. Spermiogenesis involves the changes from the spermatids internally and externally in their structural makeup. In both the males and the female reproductive system, we see mitosis and meiosis differ significantly in the terms of the events that take place, particularly in the nucleus, when we talk about the pairing of the 23 chromosomes of the two gametes. Somatic cells contain 23 pairs of chromosomes, and each pair consists of one chromosome divided by the father and another one that's divided by the mother. Mitosis is a part of the process. Again, as I said earlier, it involves somatic cell division, producing two daughter cells. Each one contains an identical pair of chromosomes. The daughter cells will contain both members of the chromosomal pairs, which are 46 chromosomes in total. Now, these cells, the new daughter cells, are referred to as diploid cells because they have now the 46 chromosomes, 23 from the mother, 23 from the father. In meiosis, it involves two cycles of cell division, meiosis 1 and meiosis 2. I don't ask you a lot of questions about meiosis 1 and meiosis 2. I do want you to appreciate the concept of meiosis being involved in the reproductive process, where you'll see the combining of the 23 chromosomes from the male and female cells to produce the 46 chromosome daughter cell. But I'll just go through the process just so you're somewhat familiar with it as it involves the reproductive system. Meiosis involves, again, two cycles. Meiosis 1 and meiosis 2 produces 4 cells, each of which has 23 individual chromosomes. The cells contain only one member of each pair of the chromosomes, and thus they're called haploid cells. Now remember, when they're combined, they're referred to as diploid cells, so you should know those two definitions, diploid and haploid, what they refer to. The event in the nucleus are the same as the formation of the spermatozoa or in the ova. And this is the process. Primarily what we're demonstrating here in the process is a chromosomal duplication that takes place inside the nucleus, differs significantly than what we saw in mitosis earlier. DNA replication also has to take place when the cells begin to the process of meiosis. DNA replication occurs within the nucleus. We see this similarly, the processes that occur in prophase that we talked about earlier in the semester when we talked about mitosis. So we see this similarly will continue in prophase. Prophase 1 arrives. Chromosomes will condense. This is visible under the microscope, and remember, in this process cells have to bring in additional energy because it requires energy for cells to undergo division, especially in the nuclei. So you'll see an increase in the cytoplasm outside the nucleus, increase in the number of mitochondria, protein products that are involved in this process. As you saw in mitosis, each chromosome consists of 2 duplicate chromatids. Then we'll see close similarities between meiosis and mitosis in this process. In meiosis, the corresponding maternal and paternal chromosomes now will join together in an event known as the synapsis. The synapsis involves the 23 chromosomes. Each member from each, the male and female pairs, will consist of 2 chromatids. These matched sets of 4 chromatids are then called a tetrad. And then there's some exchange in genetic information. This is how our characteristics will begin to exchange between characteristics of both the male and characteristics of the female will then be seen in the daughter cell from this process of exchange. The process of exchange is referred to as crossing over. Not that you're going over into the median of the dead, but the crossing over. You seen that show, Crossing Over? Yeah, okay, maybe not. Anyway, the process of crossing over just increases genetic variation because you get a little bit of the characteristics from both the male and the female. Then we go through those phases that, again, we talked about earlier in this semester, anaphase and telophase. Anaphase proceeds. In that process, maternal and paternal components are randomly and independently distributed. And then in the tetrad split, the daughter cells will receive either copy of the maternal chromosomes. As a result, telophase ends with the formation of 2 daughter cells. These 2 daughter cells contain combinations of both maternal and paternal chromosomes, but they're still a bit unique in their own characteristics because they've now picked up genetic information from both the male and the female. The distribution is variable, so the exact physical characteristics that are displayed in the daughter cell different from either parent, but unique to that individual cell. So both cells will then contain 23 chromosomes, and then this is the first process of meiotic division, which reduces the number of chromosomes from 46 to 23. This is called reductional division. Each of the chromosomes will consist of 2 duplicate chromatids, and as these duplicates will separate at the start of meiosis 2. And then in the process of interphase, which separates meiosis 1 and meiosis 2, you see no DNA is replicated during that period of time. And then we're going to come to a point at which we'll see these duplicate chromatids will then separate. It's going to yield 4 cells which will contain 23 chromosomes. And the number of chromosomes hasn't changed. So at this point, because the chromosomes, the total number of chromosomes haven't changed, it's referred to as equational division, whereas before we had reductional division. And then as the chromosomes become evenly distributed amongst the 4 cells, in males, meiosis produces 4 immature gametes that are identical in size. In females, meiosis produces 1 huge ovum and 3 tiny ovums. If you remember, last week we talked about the atrophy that occurs in those 3 tiny cells that only 1 functional ovum, referred to as the egg. The others are referred to as polar bodies. They're non-functional, and they will basically go through atrophy and will not fully develop. So a little difference in what you see in the male and the female. In the male, all of the sperm that go through, the spermatids that go through this process of spermiogenesis will develop, whereas in the female, only 1 of the eggs will develop. In spermiogenesis, or spermatogenesis rather, mitotic divisions of each of the diploid spermatogonia will produce 2 daughter cells. One of the spermatogonia will remain in contact with the basal lamina. That's the tissue layer, if you remember, epithelial tissue lying closest at the base of the cells. It will be displaced toward the lumen. Meiosis will begin, and each primary spermatocyte will then contain 46 individual chromosomes. Then at the end of the process, this is referred to as the secondary spermatocyte. And every secondary spermatocyte will have 23 chromosomes, each of which will consist of pairs of duplicate chromatids. I know I'm getting kind of wordy today. I'm not going to ask you to talk through this process. Again, I'll just bring up some key points that you need to be able to recognize. As the secondary spermatocyte then soon arrives into prophase 2, and then the completion of metaphase 2, anaphase 2, telophase 2 will yield again a grouping of spermatids, haploid spermatids, which each contain 23 chromosomes. Now all of this event occurs in the seminiferous tubules, which is the take-home point. spermatogenesis, these processes take place in the male reproductive system in the seminiferous tubules. And these illustrations are out of your book. It just goes through the steps of development of the primary, secondary spermatocytes, then eventually the haploid spermatids, and eventually the spermatozoas, which are haploid. Have to be haploid because when they reach the female reproductive system, they'll join with the female egg, which was also haploid. The 23 and 23 chromosomes will join together and form the developing embryo. Meiosis produces gametes that contain half the number of chromosomes found in somatic cells. For each cell entering meiosis, the testes produces 4 spermatozoa, whereas the ovaries will produce a single ovum. You understand the difference there. Again, all of the male sperm are going to go through development, and in the female, only one of the eggs will survive. In spermiogenesis, which is the last step of spermatogenesis, the spermatid matures into a single spermatozoa. Developing spermatocyte will undergo meiosis. Instead, we see the surrounding by the cytoplasm of what's referred to as substenacular cells. And if you recall, when we talked about the hormonal regulation in the male reproductive system, we talked about the various hormones that stimulate the production of substenacular cells. As spermatogenesis proceeds, the spermatids will gradually develop into spermatozoa. There is a process called spermiation. This is where 4 immature gametes that are of identical size, each which will develop into a functional sperm. The entire process, spermiation, takes approximately 9 weeks to occur. The substenacular cells, which are also called Sertoli cells, they play a key role in spermatogenesis. These cells have important functions directly or indirectly affects mitosis, meiosis, spermiogenesis within the seminiferous tubules. Some of their functions include maintenance of the blood-testes barrier, and we've talked about, no, that's in neurology. You haven't had neuro yet. There are several systems in our body that's based upon the tissue structure and the cells that kind of divide the body into separate chambers, and it performs a protective barrier. One is this blood-testicle barrier. Another barrier is the blood-brain barrier, where there are specialized cells inside the nervous system that help to filter out certain substances that are in our body. It won't get into our brain. There's also the placental barrier, where there are certain barriers that are set up in the female reproductive system that will help protect the baby from certain things that are in the mother's body and circulation. These filters, these specialized filters, can help protect babies, can help protect our brain, and again can help protect our testicles from toxins that may be in the blood flow, infectious agents that may be in the blood system. So these anatomical barriers that are set up inside the body are important because they help to maintain a particular safe environment for specialized functioning to occur. So this is referred to as the maintenance of the blood-testicle barrier. Seminiferous tubules are isolated from the general circulation by a blood-testicle barrier, comparable function of the blood-brain barrier. Substenacular cells are joined by tight junctions. And again, if you go back to the first part of anatomy, we talked about how cells are stuck together. They're stuck together by specialized processes, desmosomes, hemidesmosomes, tight junctions that are like the cement that holds the cells together. So we see the substenacular cells are joined together by tight junctions. They form a layer that divides the seminiferous tubules into an outer basal compartment which contains spermatogonia, and then an luminal compartment or an adluminal compartment where meiosis and spermatogenesis will occur. We also will see transport across the substenacular cells. This is a tightly regulated process, so conditions in the luminal compartment remain very stable. Again, a separation of environment. The fluid in the lumen of the seminiferous tubules is produced by substenacular cells, which will regulate fluid composition. This fluid is very different from the surrounding interstitial fluid. It's high in androgen, estrogen, potassium, and amino acids. The blood-testicle barrier is essential in preserving the differences between the tubular fluid and the interstitial fluid. In addition, this barrier prevents the immune system cells from detecting or attacking developing spermatozoa. This is important as well. because they have their cell membranes that are sperm-specific antigens found in somatic cells. The membranes thus must be identified. If they're identified as foreign by our system, then they would be attacked and destroyed, and the person would become sterile. There are some autoimmune processes that specifically target these cells, and these individuals are sterile for the rest of their lives. So it's an important process to maintain a separation there in that system. Spermatogenesis depends on the stimulation of the substanacular cells. And if you recall from last week, and again, memorize that illustration, that table that I put up that showed the hypothalamus, the pituitary gland, follicle-stimulating hormone, luteinizing hormone, and then what cells are affected, both in the male and the female reproductive system. There are probably 4 questions on the test that if you can just quickly pencil out that chart, you'll be able to answer the questions. Not only will you know where the stimulating hormones come from, you'll know where the effect inside the reproductive systems are. So in the process, we see circulating follicle-stimulating hormone and testosterone. Spermatogenesis depends on the stimulation now of the substanacular cells, and those cells are stimulated by follicle-stimulating hormone testosterone. Stimulated substanacular cells then will promote division in spermatogonia and the meiotic divisions of the spermatocytes. Spermiogenesis requires the presence of substanacular cells. These cells surround and enfold the spermatids, providing nutrients and chemical stimuli that will promote development. The substanacular cells also phagocytize cytoplasm. That is, it sheds the spermatids as they develop into spermatozoa. The substanacular cells will secrete peptide hormone referred to as inhibin. You should know inhibin. Inhibin in response to factors releasing by the developing spermatozoa. Now, inhibin depresses the pituitary production of follicle-stimulating hormone. Perhaps, we're still kind of figuring this out, at the hypothalamic secretions of gonadotrophin-releasing hormone. But we do know that inhibin depresses the production of pituitary follicle-stimulating hormone. That's a test question. The faster the rate of sperm production, the more inhibin is secreted. So regulating follicle-stimulating hormone and gonadotrophin-releasing hormone secretion, substanacular cells provide feedback control of spermatogenesis. And again, the maintenance of homeostasis comes into play here. Again, there's a feedback mechanism, a negative feedback mechanism, where the end product will then secrete a substance that will go back and shut off the mechanism by which the process is stimulated. So we see the faster the rate of sperm production, the more inhibin that's produced. This will, in essence, slow down sperm production because the inhibin then goes back in a feedback mechanism and depresses the pituitary function of follicle-stimulating hormone, and also in the hypothalamus where the releasing hormones are at, higher than the pituitary gland. It also will shut off production there. At least it's theorized that it will shut off production there. So we see 2 influences of inhibin that's very important in maintaining homeostasis inside this productive system. Then secretion of androgen-binding proteins. Androgen-binding proteins bind androgens, primarily testosterone, and fluid content of seminiferous tubules. This protein is thought to be important in both elevating the concentration of androgens within the seminiferous tubules. The production of androgen-binding protein is stimulated by the follicle-stimulating hormone. There's also another substance referred to as Mullerian inhibiting factor. Mullerian inhibiting factor is secreted by the substanacular cells in the developing testicles. Hormone causes regression of fetal Mullerian ducts. These are the passageways that participate in the formation of uterine tubes and the uterus in females. In males, inadequate Mullerian inhibiting factor production during fetal development leads to the retention of these ducts and failures of the testicles to descend into the scrotum. I think that's the test question for Mullerian inhibiting factor, was about the substance that keeps the testicles from descending. And you remember when we talked about the descent of the testicles, right, in the retro-abdominal area, descend and come through the inguinal canals. And so Mullerian inhibiting factor is felt to lead to a failure of the testicles to descend into the scrotum. during fetal development. So you have to have an adequate amount of Mullerian inhibiting factor. Now a little bit about the anatomy of the spermatozoon. It's three distinct regions: a head, a middle piece, and a tail. And on your, I don't know if I'm going to have that on the lab exam this year or not, but I had the sperm up there before. There was something about the illustration that caused a lot of confusion. But my point was to ask you to identify the head, the middle piece, and the tail on the sperm. I would look at that illustration, but I may not put that on the exam this week. Not this week, sorry. Not to cause any panic. That's for your final. At the tip of the head is what's called a chromosomal cap. This is a membranous compartment that contains enzymes that are essential to fertilization. I believe that's a test question as well. A chromosomal cap, these enzymes are very important because when we talk about the female process here of development, you'll see that the ovum is actually covered by a layer of cells, and those cells actually act as a protective barrier for the egg. And so for fertilization to take place, these enzymes have to get in there and break down those cells to be able to attack. This illustration here is out of your textbook, and it shows the different regions of the spermatozoon. This is the chromosal cap that I referred to, area that's packed with enzymes. Another little view that you can see a little bit better, the chromosomal cap. In the process of spermiogenesis and spermatozoon, we see differentiation of the spermatid into spermatozoon. The differentiation process takes about 5 weeks. There's a short neck that's attached to the head of the middle piece. The neck contains both centrioles of the original spermatid. The microtubules of the distal centriole are continuous, and those of the middle piece of the tail. The mitochondria in the middle piece are arranged in a spiral around the microtubules. The mitochondrial activity provides ATP required to move the tail. The tail is the only flagellum in the human body. A flagellum is a whip-like organelle that moves the cell from one place to another. We see cilia which beat in a predictive wave-like fashion. The flagellum of the spermatozoon has a complex corkscrew motion. You should know that about the flagellum. The flagellum, the whip-like organelle, it moves the cell from one place to another. It's the only flagellum in the human body, so it's not hard to keep up with. Spermatozoan structure, we see that it lacks endoplasmic reticulum, it lacks a Golgi apparatus, lysosome, peroxisomes, inclusion. Many of those intracellular structures that the rest of our cells do have. The loss of the organelles reduces the cell size and mass, and so it's essential as a mobile barrier. The enclosed chromosomes, the extra weight would only slow it down. Sperm have to move pretty fast. Because the cell lacks glycogen or other energy reserves, it must absorb nutrients, primarily fructose, from the surrounding medium. And I think fructose is on the test as well, if I remember correctly. Something about fructose being the energy reserve. Spermatogenesis begins at puberty, continues until relatively late in life, and in men you can see it extends to around the age of 70, some a little bit longer, some a little bit less. It's a continuous process at all stages of meiosis, and it can be observed within the seminiferous tubules. We see hormonal interaction. The hormonal interaction regulates the reproductive function. The anterior lobe of the pituitary gland releases follicle-stimulating hormone. Remember, gonadotrophin-releasing hormone stimulated from the hypothalamus is its trigger. And also releases luteinizing hormone, so FSH and LH. The pituitary release of the hormones, luteinizing hormone and follicle-stimulating hormone, in response to, I'm being redundant now, gonadotrophin-releasing hormone. Gonadotrophin-releasing hormone is a peptide synthesized by the hypothalamus and carried to the anterior lobe of the hypophyseal portal system. This is the circulatory system. Remember, in the endocrine system, I'm confusing classes again, I'm sorry. A&P2, it just got through doing endocrine system, so you don't know this, so I'm sorry. Well, you may know this. I shouldn't assume anything, right? In the endocrine system, it's a very vascular system. In the endocrine system, it's similar to the nervous system in the effect that you'll see on the cells, but there are chemical substances, hormones, that are produced inside our body that enter into the bloodstream and carry out their response. So whereas a nerve sends an electrical impulse and it happens right away, the endocrine system sends a chemical messenger. So it secretes a hormone, like luteinizing hormone and follicle-stimulating hormone. And the hypophyseal portal system is the blood supply that's for the endocrine system. So we see that in the hypothalamus and the pituitary gland will secrete their substances into this hypophyseal portal system. So that's what I mean by entering into the bloodstream at that system. When you do the endocrine system next semester, it'll make even more sense. But as you look at your illustration, when you see hypothalamus and you see pituitary gland, hypothalamus first, gonadotrophin-releasing hormone, pituitary gland, it lies under the hypothalamus. You'll see the secretion of gonadotrophin-releasing hormone, luteinizing hormone, follicle-stimulating hormone. These messengers have to get one place to another, and that goes through the hypophyseal portal system, which will then contact the rest of the circulatory system. And so if there are other target organs, such as the testicles and other locations of the body, these chemical messengers are now in the bloodstream and have specific receptors at the target organ that will pick up those chemical substances that have been released. Okay? A little quick overview of the endocrine system there. This is the illustration I was telling you about again. And as you can see, the hypothalamus lying in the most superior of positions. And I believe there's also, and if it's not, it won't hurt you to flip through your book to the endocrine system and look at a specific anatomical picture of the hypothalamus and the pituitary gland. Pituitary gland is divided into two parts: anterior pituitary, which has three portions, and then the posterior pituitary gland. We're talking primarily about the anterior pituitary gland. And then again, in the hypophyseal portal system, as these substances are being released, that has to get to the testicle, so it goes through the circulatory system, and then it implies its influences on the interstitial cells, one for the secretion of testosterone, seminiferous tubules for secretion of substenacular cells, which will in turn, and remember when we talked about the process of spermiogenesis, as we start to produce more sperm, we get the secretion of, from the substenacular cells, we get the secretion of inhibin. And inhibin in a feedback mechanism will go back and inhibit follicle-stimulating hormone, will go back and is theorized will also inhibit the hypothalamus, which will then shut off that process. Again, in the process where the substenacular cell is important in the producing of androgen-binding hormone that we talked about in the stimulation of spermiogenesis. We also see here for testosterone, when the interstitial cells, and again, if you can draw out this chart real quickly, you see the interstitial cells important in the secretion of testosterone. And what does testosterone do? It has an effect on the central nervous system, stimulation of bone and muscle growth. We also see establishing and maintenance of male secondary sexual characteristics, and then accessory glands and organs as well. Male pattern hair growth, all those types of things. Talk a little bit about the hormones quickly. Gonadotrophin-releasing hormones secreted in pulses rather than continuously. In adult males, small pulses occur 60 to 90-minute intervals. As the levels of gonadotrophin-releasing hormones change, so does the rate of secretion of follicle-stimulating and luteinizing hormone, which only makes sense. We only get follicle-stimulating and luteinizing hormone when we get gonadotrophin-releasing hormone secreted. Testosterone is released in response to luteinizing hormone, as I showed you in the illustration before. And again, if you know this chart, if you can draw this chart out, you'll be able to answer at least four questions. It may even be 5 questions on the test just off this chart alone. So you should be able to do that. Whoops, sorry. Unlike the situation in women, gonadotrophin-releasing hormones pulse frequency in adult males remain relatively steady from hour to hour, day to day, year to year. We'll talk about what goes on in the female system next week. But as a result, plasma levels of follicle-stimulating hormone, luteinizing hormone, testosterone remain within a relatively narrow range until relatively late in life. In the males, follicle-stimulating hormones targets primarily substenacular cells and the seminiferous tubules. Under follicle-stimulating hormone stimulation, in the presence of testosterone in the interstitial cells of the substenacular cells, it promotes spermatogenesis. We also see the secretion of androgen binding proteins. The rate of spermatogenesis regulated by the negative feedback mechanism that we already talked about, the production of inhibin as it goes back to the level of the pituitary gland and possibly back to the level of the hypothalamus to shut off the system. Under gonadotrophin releasing stimulation, follicle stimulating hormone promotes spermatogenesis along the seminiferous tubules. Spermatogenesis, again, remember, takes place in the seminiferous tubules. Again, I think that's a test question. Spermatogenesis accelerates, however, so that the rate of inhibin secretion by the substernacular cells of the testes. Inhibin inhibits follicle-stimulating hormone production in the anterior lobe of the pituitary gland, may also suppress the secretion of gonadotrophin in the hypothalamus. Net effect is that follicle-stimulating hormone levels become elevated. Inhibin production increases with follicle-stimulating level to return to normal. If follicle-stimulating levels decline, inhibin production falls, so does the rate of follicle-stimulating hormone production accelerates. In males, luteinizing hormone induces the secretion of testosterone, definitely a test question, and the other androgens by the interstitial cells of the testes. Testosterone is the most important androgen. 4 functions of testosterone, and I think this is a question as well. Stimulating spermatogenesis and promoting functional maturation of the spermatozoa through the effects of the substernacular cells, so it stimulates spermatogenesis. We also see it affects the central nervous system, which includes libido, sexual drive, and related behavior. We also see it stimulates metabolism throughout the body, especially the pathway concerned with protein synthesis, blood cell formation, and muscle growth. We also see establishing and maintaining male secondary sex characteristics such as the distribution of facial hair, increased muscle mass, body size, and the quantity and location and characteristics of adipose tissue, maintaining the accessory glands of organs in the male reproductive system. Testosterone functions like all other hormones, circulating in the bloodstream while bound to 2 types of proteins, gonadal steroid binding globulin, and I'm not going to ask you a question on these, just for FYI. Gonadal steroid binding globulin, and then the albumins, which bind the remaining 1/3 of the testosterone. Testosterone diffuses across the cell membrane on target cells and binds to intracellular receptors. These receptors are at the target organs. The hormone receptor complex then binds to DNA in the nucleus, many of the target tissue, some arriving from testosterone. It converted into dihydrotestosterone, or DHT. Small amounts of DHT diffuse back out of the cells into the bloodstream. Levels are usually about 10% of the circulating levels of testosterone. Now, DHT can also enter into peripheral cells and bind the same hormone receptors targeted by testosterone. In addition, some tissue, notably that of the external genitalia, will respond to DHT rather than testosterone, and other tissues, including the prostate gland, are more sensitive to DHT than testosterone. Thus, testosterone production begins around the 7th week of fetal development and reaches the prenatal peak at around 6 months. We also see other factors. The Mollarin inhibiting factor that we talked about a little bit earlier, by development, substernacular cells will lead to a regression of Mollarin ducts. The early surge in testosterone levels stimulates differentiation of the male duct system and the accessory organs, and it also affects the development in the central nervous system. The best known effect on CNS, the central nervous system, occurs in development of the hypothalamus. Testosterone apparently programs the hypothalamic centers to be involved with gonadotrophin releasing hormone production and the regulation of follicle stimulating hormone and luteinizing hormone at the level of the anterior pituitary gland. We also see influences sexual behavior and sexual drive. As a result to prenatal exposure, testosterone, the hypothalamic centers will respond appropriately when individuals become sexually mature. The factors responsible for regulating fetal production of testosterone are not clearly known at this point. Testosterone levels are low at birth until puberty. Background testosterone levels, then we'll see the background relatively low, higher in males than in females. Testosterone secretion will then accelerate markedly during puberty, initiating sexual maturation and the appearance of secondary sexual characteristics. In the adult male, there is still negative feedback control to maintain homeostasis in the level of testosterone production. Above normal testosterone levels inhibit the release of gonadotrophin by hypothalamus, causing reductions in luteinizing hormone and lowering testosterone levels. The plasma of adult males also contains relatively small amounts of estradiol, much lower levels obviously than in females. 70% of estradiol is formed from circulating testosterone. The rest is secreted primarily by the interstitial and the substenacular cells of the testes. Conversion of testosterone to estradiol is performed by an enzyme known as aromatase. Unknown reason, estradiol production increases in